Welcome all to the today's session in the genetic module. So today we will discuss about the pedigree analysis. So pedigree analysis. What is pedigree? It is the pictorial representation of a family tree. It shows the inheritance of one or more trait across the generation. Okay. So what are the aims of pedigree analysis? It determines the mode of inheritance. It predicts the risk to the offspring. It assigns the genotype. It study gene versus environment contribution and identify the carriers of the disease. So the most important from examination point of view, it is the mode of inheritance and predicting the risk of the offsprings. Okay, these are the two things which are more commonly tested in the examination. So what we are going to learn, construct the pedigree using standard symbols. Okay, so identify whether it is autosomal dominant, whether it is autosomal recessive, whether it is X-linked, either dominant or recessive, and we should be able to calculate the recurrence risk and recognize the limitations of the pedigree itself. Okay, so what are the basic terminologies which are used in the pedigree analysis? It is a proband. So what is proband? It is the first affected individual which is studied. Okay, that is marked by arrow. Okay, so these terminologies sometimes they can ask you in the SBA. The proband is called as, and they will give you multiple options. The proband is the first affected individual which is studied. Okay, then consultant. This is the person seeking the genetic advice. Okay, consultant. That is the person seeking the genetic advice. Then sibship. That is the brothers and sisters. Then generation. Each level in the pedigree is called a generation. Okay, you have to study minimum 3 generations so that you get a fair idea. Okay, so that is what is some basic concepts about the pedigree analysis. Then what are the standard symbols which are used in the pedigree? Square for male, circle for female, shaded if it is affected, then we show as a shaded, either square or the circle. Half shaded is the carrier. Horizontal line is the marriage. Okay, vertical line denotes the offspring. Arrow is the proband. Okay, so these are the standard terminologies. Okay, so normal female is by circle, normal male is by the square. Okay, if the sex is not known, then it is shown like this. Okay, sort of diamond. Okay, here horizontal line is the marriage. Okay, when there is a mating between the relatives, here it will be double line. Okay, sort of consanguineous marriage. Okay, so this is the offspring. Vertical line will be the offspring. Okay, so this is the first sibling, this is the last born. That is how you have to denote. Okay, so sex unknown with phenotype of interest, so this will be completely darkened. Female heterozygous for recessive allele, half is shaded. Okay, male heterozygous for recessive allele, half is shaded. When there is a stillbirth or spontaneous abortion, full is shaded. Okay, this is the way the disease is shown. Okay, if the person is diseased, then it is shown like this. Okay, so the proband will be shown like this with the arrow. Okay, so these are the few symbols. Sometimes they can ask you in the SBS, so I have just covered it. Okay, then key principles of pedigree analysis. It is always a stepwise approach. Look at the generation pattern. Okay, so when you will have 3 generation minimum, so look at the pattern in their particular generation. Okay, check sex distribution. Most important thing, which are the sex which are getting affected, whether all males are getting affected, how is the transmission. Look for male to male transmission. Look for skipped generation. This is very specific. Okay, you have to look for skipped generation. Then assign possible genotypes and confirm the mode of inheritance. Okay, so when you solve these pedigree questions, you should always look at the generation pattern, check the sex distribution, look for male to male transmission, look for skipped generation. So these are certain things which we will further go into the details in the subsequent session. Okay, so uh, let us talk about patterns of inheritance. How does the autosomal dominant and autosomal recessive they behave? Okay, so autosomal dominant will be seen in every generation. Remember, there is a no skipping of the generation in autosomal. Every generation you will see the disease. Okay, while in autosomal recessive, a particular generation will not have the disease. So there is a skipping of the generation. Then we are likely to deal with autosomal recessive. Okay, as I mentioned, you see 3 minimum generations. And if you see that the generation is getting skipped, you are likely to deal with autosomal recessive. Then parents. Affected parent will be present always in autosomal dominant, while parents are often normal in autosomal recessive because there is a skipping of the generation. Sex ratio, both male and females will get affected. Here, both males and females will get affected. Transmission, male to male is present in both autosomal as well as dominant as well as recessive. Example, pattern is vertical while it is horizontal. Okay. Now coming to the X-linked. Okay, how does X-linked recessive and X-linked dominant behave? Okay, so here in X-linked recessive, males are more commonly affected, while X-linked dominant, both sexes are affected. Females are affected. X-linked, it is not seen. Females are not affected in X-linked recessive. However, in the X-linked dominant, females are affected. Male-to-male transmission is absent in X-linked. It is also absent in the X-linked dominant as well as recessive. Carrier female is always there, while here there are no carrier females in X-linked dominant. Skipping of the generation is seen with X-linked and it is not seen with the X-linked dominant. So when we talk about skipping of the relation, it will be either X-linked recessive or it will be autosomal recessive. Okay. Now coming to the genotype assignment, how the genotypes are assigned. So when you have autosomal dominant, the affected is denoted as AA. Okay. So even in homozygous, heterozygous state, this will have the affection. Okay. Even if there is a small a which is present, then also, so autosomal dominant is shown as affected as AA. When we are dealing with autosomal recessive, the affected will be shown as a small a, because when there are both alleles which are present, then only it is getting expressed, while the carriers will be shown as AA, capital A, small a. I hope you are getting this. Okay, so that is how the genotype is assigned in autosomal dominant and autosomal recessive. What happens in the X-linked? The male hemizygous will be shown as XAY. Okay, so that is the male hemizygous. Okay, now coming to the risk calculation. If it is an autosomal recessive carrier present, then there is a 25% chance of affection in the offspring. Okay. If it is an autosomal dominant heterozygous parent, then there is a 50% chance of the affection of the offspring. If it is an X-linked carrier state, there is a 50% of the sons will get affected. When the X-linked carrier mother is there, the 50% of the son will get affected. Okay. So what are the applications of pedigree analysis? To identify the inheritance pattern, genetic counseling, estimate the recurrence risk, detect the carriers, and study rare genetic disorders. Now, what is the importance? It is a non-invasive method. It is useful in human genetics, helps in clinical decisions. Okay, so coming to the predicting mode of inheritance, there are certain core principles which you should remember. Pedigree analysis helps to predict the most likely inheritance pattern. So you can get the most likely pattern just if you study the pedigree. Okay. So it is based on the generation pattern, sex distribution, and transmission clues. Okay. There are 5 basic Mendelian patterns which you are going to see. What are those 5 basic patterns? One is autosomal dominant, second, autosomal recessive, third, X-linked recessive, fourth, X-linked dominant, or it can be Hollandric, that is Y-linked. Okay, so these are the 5 types of dominant, and what will be the pedigree in each, that we should be able to understand. Okay, so let us see the key clues, key rules here. Okay, so when we talk about Mendelian traits, Mendelian trait means it is the single gene disorder. Okay, so all single gene disorders will follow this Mendelian trait. Okay, so this is the rule which you should remember, whether it is autosomal chromosome or whether it is a sex chromosome. If we are discussing about single gene disorder, it has to follow the Mendelian trait. Okay, so it includes autosomal as well as sex linked chromosome. So it doesn't mean that the Mendelian trait is only for autosomal. Okay, it includes for both, but it has to be single gene disorder. Okay, so even traits on sex chromosomes are still considered Mendelian. Okay, So autosomal dominant. First, we will talk about autosomal dominant. It does not skip the generation if fully penetrant. Both sexes are equally affected. That means male are affected, female are also affected, and there is a vertical transmission pattern. See this pedigree. Okay, so here total 4 generations you are able to see, or this is first generation, this is second generation, this is third generation, this is fourth generation which they are seeing. Okay, so there is no skipping of the generation. Generation is not skipping. This is present, this is present, this is present. Okay, here disease is absent, but the parents does not have the disease. Okay, if the parents are affected, the next generation is always affected. Parents are not affected, the next generation will not affect. But there is no skipping of the generation. Once there is a disease affected person, there is no skipping of the generation, and both males and females are affected. So autosomal dominant, it will not skip the generation. It affects both sexes equally, and vertical transmission pattern is seen. Okay, so diagnosis often made by excluding other patterns and helps to increase the confidence in interpretation. Okay, so important thing here is the father-son transmission is present. Okay, so father can transmit the disease to the son. Okay, so father can transmit the disease to the son. Here is a father who can transmit the disease to the son. Okay, which is not seen in X-linked. Okay, then only males are affected, likely X-linked recessive. Okay. Then skip generations, likely recessive, either X-linked or sex-linked. Okay. Either X-linked recessive or autosomal recessive. There is a skipping of the generation. If there is affected father plus unaffected daughter, we are not dealing with X-linked dominant. Okay. If there is affected father and there is an unaffected daughter, then we are not dealing with the X-linked dominant. So these are the certain clues you should remember when you are solving the questions about pedigree analysis. Father to son transmission is there. That means it is not a X-linked. Okay. In X-linked, mother will be carrier and son will be affected. Okay. And there is no father to son transmission. Only males are affected. That is suggestive of X-linked recessive. Okay. Then skipping of the generation, it is recessive, either X-linked or autosomal. Then affected father plus unaffected daughter, that is also not a X-linked dominant. Okay. Now let us see further. Son gets X chromosome only from the mother. Father cannot pass X-linked trait to the son. Okay. So X-linked trait, father will never transmit to the son. So if there is a father to son transmission, that means it is not a X-linked. So this is father to son transmission is not X-linked. Okay, this is how we should rule it out. Okay, so look at the pedigree, whether the males are getting affected, whether the females are getting affected, whether the father is transmitting to the son or not transmitting to the son. This is all you have to look at the chart and then interpret. Okay, therefore male to male transmission rules out X-linked inheritance. Okay. Now, next is a X-linked dominant clue. Okay, we have just seen X-linked dominant. Okay, here there is affected father. All daughters will be affected, and if daughters are unaffected, it is not a X-linked dominant. So look at the affection. If the father is affected, all daughters are always affected. Okay. Here there is no father affection, so all daughters are not affected. Here father is affected, so all the daughters are affected. Okay. If daughters are unaffected, it is unlikely to be X-linked. Okay. So that is how you should classify. Okay. So there are certain limitations, practically speaking, when you solve this, because when you have a very small family size, the pattern is not very clear. When you have a large family size, you put each and every member there, then you will get a better idea. But because of small family size, sometimes the pattern will not be very clear. Then incomplete records. If there are missing links, that is also limitation. Then reduced penetrance. Trait may appear to skip. Sometimes certain traits will skip. Okay, then variation in expression, the severity will differ. Okay, so that may put us in classifying or putting into the pedigree chart. There is a minor severity which has not brought to the medical attention, so clinically not significant, but trait is there. Okay, so these are certain limitations. So key conclusion: not all features of a pattern are always present. Therefore, always conclude most likely mode of inheritance. Okay, we cannot say absolutely, but most likely mode of inheritance. Okay, so from examination point of view, pedigree analysis is the pattern recognition plus exclusion. That is the key principle which we will follow. Never rely on a single feature. Always combine generation pattern, sex distribution, and transmission clues. Okay, so these are the things which you should remember. So just quick algorithm: if it is a vertical pattern, it is a autosomal dominant. If it is skipping the generation, that means it is likely to be autosomal recessive. Only males are affected, that means it is a X-linked recessive. If father to son transmission is present, that means it is autosomal but not X-linked. Okay, all daughters are affected and fathers are affected, it is X-linked dominant. Okay. Now let us see about proband. Who is proband? Proband is the first affected individual through whom a pedigree is initiated. It is marked by an arrow in the pedigree chart. Okay. Also called as index case or case of ascertainment. Okay. Now we will see the individual pattern. That is the autosomal dominant trait. So basic concept here is that trait is expressed in heterozygous state also. If it is a dominant trait, it will be getting expressed even in the heterozygous state. That is, even if capital A small a is there, it will get expressed. In homozygous AA, that will be lethal and that is rare. Okay. So key characteristics is that there is no skipping of the generation, always vertical transmission, parents affected individually usually has affected parent. So affected individual will always have a affected parent. Okay. Sex distribution is seen both males as well as females will get affected. Transmission is seen in both sexes. Male to male transmission is also present, and penetrance may appear skipped if incomplete penetrance. So this is, in certain cases, it may appear skipped if there is an incomplete penetrance of a particular gene. Okay. But generally, there is always affection of both the sexes and there is a vertical transmission. Okay. Now calculating the risk. So if this is a heterozygous affected person, AA marries with AA. Okay. There is a risk of affection is 50%. Okay. If both parents are unaffected, unlikely autosomal dominant, because in that particular case, the possibility is that there is a new mutation which has happened. If it is autosomal dominant, at least one person will get has to be get affected. But if both parents are not affected, but the offspring is affected, in that particular case, we will think of new mutation. Okay, so new mutation possible during the gametogenesis. So examples of autosomal dominant are neurofibromatosis type 1, familial hypercholesterolemia, Marfan syndrome, brachydactyly, and phenylthiocarbamide testing. Okay, so these are the disorders of autosomal dominant in nature. Next is the autosomal recessive trait. What happens in the autosomal recessive? So trait are expressed only in homozygous trait. So when it is autosomal recessive, the both alleles should be present. Okay, so it will be present only in homozygous state. So the trait is expressed only in homozygous state when both alleles are present. Okay, so parents will be usually carrier, that is capital A and small a. Okay, so here you see one of these person is heterozygous. So either this is heterozygous or this is heterozygous, and that is transmitting the further to the next generation. Okay, so here it is heterozygous. Okay, now here there is a consanguineous marriage and here there is affection of the male. Okay, so autosomal recessive. So what happens in the autosomal recessive? There is a skipping of the generation. Okay, so here this appears as a skipping of the generation because one of this person is heterozygous, but it is getting expressed somewhere here. Okay, here also it is heterozygous. Okay, so generation, there is a skipping of the generation. Parents usually unaffected carriers. Parents will be unaffected carriers. There will be sex distribution will be equal. So we are dealing with autosomal recessive. So there is affection of the male as well as affection of the females also. Consanguinity, common association. Here there is a consanguineous marriage. So there is a homozygous recessive form. Okay. So risk calculation here, when one carrier will marry with other carrier, there is a 50% carrier and 25% affection rate. Okay, this you should remember. When two carriers will marry, there is a 25% chance of affected child and 50% will be carrier and 25% will be normal. So they always ask this risk calculation in the examination. Okay, then consanguineous marriage will increase the risk due to the shared alleles. Okay, so examples of X-linked, autosomal recessive is phenylketonuria, cystic fibrosis, Tay-Sachs disease, sickle cell disease, and glycogen storage disease. Okay,